Welcome. I'm Kathy O'Connor with Global Genes. Welcome to the morning session. We're really excited to have you guys here. I know the other room was pretty crowded, so they're trying to work out that stuff for now, but welcome. At least you have a table and chairs in here. So I am excited to welcome you to this morning breakout session, focusing on science and technology. We are excited that this track will be live streamed as well through Platform Q, so welcome to any of our remote attendees also. Without further delay, I would like to introduce our first session on using sequencing to understand rare idiopathic disorders. Please join me in welcoming Dr. Jeanette McCarthy, the genome educator at University of California, San Francisco, and Center for Applied Genomics and Precision Medicine at Duke University, and Caroline Loewy, the co-founder of the KCN Q2 Cure Alliance. Welcome. Here.
Hi, everybody. It's a pleasure to be here today. What I'd like to do in the next 15 minutes or so is really take a look under the hood of using next-generation sequencing for solving undiagnosed genetic diseases, rare diseases in particular. So can I just have a show of hands? How many of you have had experience with this for a child or somebody that you know having undergone genome sequencing? Great. Excellent. So I would really like to approach this from a lay perspective. I have experience solving diagnostic dilemmas using sequencing, but I really want to bring it down and help you understand what goes on underneath the hood there. So it starts with understanding a little bit just about genetics and human biology, and I'm sure you're all quick studies on this now, having undergone sequencing. But basically, when you think about the human body, it's really remarkable. We're made of trillions of cells, and each of those cells does something different. So the cells in your pancreas, for example, produce a protein insulin, which helps to regulate blood glucose. The cells in your immune system make proteins, antibodies, which help recognize foreign pathogens and mount an immune response. And the instructions for making these proteins are encoded for in your DNA. And the instructions for making every single protein in your body are in your human genome. And that human genome is located in just about every cell in your body, where it's virtually identical. And that's why it's possible to draw a sample of blood from your arm, or take a scraping of cells from the inside of your cheek and access your genome. And that genome, as you know, is made of DNA, building blocks for different what we'll call letters or nucleotides, ATGC, which I've color-coded here. They're just strung together in a long strand. And when we talk about sequencing someone's DNA, we're talking about understanding the order of these nucleotides in the chain. Here we have an example of a nucleotide chain that's 34 nucleotides long. To put this in perspective, your human genome is 3.2 billion nucleotides. Now, if you think about that whole human genome, that 3.2 billion nucleotides, we actually know relatively little about what it does. We know the most about the regions of the genome that code for proteins, and these are your genes. But that only represents less than 2% of your genome. There are about 22,000 human genes. The remaining DNA we call non-coding DNA, and honestly, we know very little about what that's doing, but there's ongoing efforts to figure that out. Human beings are 99.9% identical at the genetic level, but that means at about 4 to 5 million nucleotide positions in our genome, on average, we differ from one another. And that type of variation can take many different forms. The most common type of variation you see are very small changes in the genetic code. Things like insertions or deletions of just a 1, 2, or 3 nucleotides, for example. You have single nucleotide substitutions where the wrong nucleotide is added into the chain. These are by far the most common type of variation, but they occur all over your genome, not just in genes, but mostly in the non-coding region, that 98% of your genome. If they do occur in genes, it doesn't necessarily mean that they're going to alter the gene or the protein product of that gene. They can, so sometimes you'll get even a single nucleotide change that results in a change in the length of the protein, represented in blue here, so that now you have, for example, a shorter protein, and that protein then loses its function. It can't do what it's supposed to do. You have single amino acid changes that can occur, and these result in, as they sound, a change in the amino acid from one to another, and these may or may not affect the protein. Sometimes they can cause diseases, other times they're completely benign. And then you can have changes at the level of DNA in genes that have absolutely no effect on the structure of the downstream protein. This is what an average human looks like. We're all walking around with about 4 to 5 million variants. About 55,000 of these occur in our genes. About 100 of these cause a loss of function of the protein. And about 10,000 of these are these missense or single amino acid change variants. That's the average human being. But once in a while, you have a human being who ends up having a more serious genetic aberration, something like duplication of an entire chromosome, so 3 chromosomes instead of 2, as is in the case with Down syndrome, where you have 3 copies of chromosome 21. Sometimes you have a case where an entire chunk of DNA from one chromosome breaks off and switches places with another chunk on another chromosome. And sometimes you have large chunks of DNA in your genome that are completely deleted or duplicated in tandem. Now, fortunately, these types of larger genetic variations are quite rare because they are often very, very deleterious. So one of the things is we have 4 to 5 million variants in our genome, and certainly they're not all bad. Think about mutations and variation as being just a natural phenomenon. We all have lots of variation, but the real question is which of these mutations or variations actually cause disease? And when you think about genetic disease, you really need to think about it as a spectrum. You have on the one hand diseases or genes where a single gene is sufficient, a mutation in a single gene is sufficient to cause the disease. These are purely genetic diseases. On the other hand, you have purely environmental diseases, things like influenza or bee sting or something like that. And in between, you have all of these complex genetic diseases due to the combination of genes and environment. I'm going to focus on the first category, the purely genetic diseases, what we call Mendelian genetic diseases. These classical Mendelian diseases are, again, ones where a mutation in a single gene is sufficient to cause the disease. There have been about 8,000 rare diseases described. About 80% of those are thought to be Mendelian genetic diseases, and genes have been identified for about 4,500 of these. And it's really fascinating because I started my graduate work in the late 1989, I think it was, and just the progress we've made identifying the genetic basis of the diseases is incredible, and it's only accelerating as we've been applying next-generation sequencing techniques. So let's turn our attention to diagnostic sequencing. So this is what many of you are familiar with. This is using next-generation sequencing to help diagnose a rare disease in a child, for example. What we're really trying to do is ask, in this person who has about 4 to 5 million variants, how can we filter through all of those variants and come up with that one variant that causes disease? And so what we do is we apply some very logical filters. The first thing we might ask is, well, what do we know about the genetic basis of Mendelian diseases already? We know that the majority of the variants that cause these diseases are located in genes. In other words, they're not in that non-coding region of the genome. They're actually in genes. We call this region of our genome the exome. And in fact, sometimes you may even start at this point. Instead of doing whole genome sequencing, you do whole exome sequencing. What else do we know? Well, we know that these diseases are rare, incredibly rare, which means that the genetic variants causing them are going to be even rarer. We also know that these variants, when they occur in genes, are likely to alter the protein in some way, alter the structure of the protein. So we filter, again, not even taking the disease into account, just knowing that variants are rare and protein altering can bring us down to about 1,500 variants in our genome on average. Then we can also take advantage of other family members. If you have, for example, a family where you have an affected individual with the disease and they have a sibling who also has that disease, you know that those siblings should be sharing the causal variant. And so you can filter even more based on that and get down to about 1,200 variants. So you're left with 1,200 variants, maybe even in 1,200 different genes in your genome, and you need to figure out which one of these is causing my disease. So the other things we can do is ask, has this variant been seen before? Has it been seen before, this exact same variant, in healthy people? And we have resources that we can use. We have variant databases. The 1,000 Genomes Project, EVS, and EXACT databases are publicly available resources where these investigators have sequenced the genomes of tens of thousands of individuals and placed all of that information in the public domain. You can imagine if you have a child with a very rare genetic disorder and that variant is very deleterious, you're not going to see it in these populations because these are ostensibly healthy people. So we can filter out a bunch of variants that way. But we also would want to know, has this exact same variant been seen in the context of disease? In particular, the disease that your child has. And for this, we turn to other databases. For example, the open access database ClinVar, which is a repository where investigators and clinical laboratories and other people can deposit information about specific genetic variants that they found in the context of disease, and you can search and see if your variant is in here. There's also a commercial option called the Human Gene Mutation Database. So if your exact variant hasn't been seen before, you might find that the gene that that variant is in has been described before and associated with disease. In other words, other variants in that gene may have been described for disease, and you can assess this by looking at databases such as OMIM, Online Mendelian Inheritance in Man. You could search there, for example, if I have a mutation in a gene BBS1, I can plug it in and it'll pull up and show me that, oh, this gene, mutations in this gene have been associated with Bardet-Biedl syndrome. And the great thing is you can drill down even further and it will list all of these different clinical manifestations that have been observed in patients with mutations in this gene. Things like dysmorphologies of the head or other body parts, skeletal system, neurological abnormalities, things like that. So you can really drill down and look at what we call the phenotype. So if you have a variant and it hasn't been seen before, and maybe it hasn't been reported in a gene that's known to cause disease, you're left with what we call research candidates. So these are the ones that leave you scratching your head. But it's not all lost because there's other information that can be brought to bear to help determine if these research candidate genes are in fact underlying your disease. One is asking whether the gene is very similar to another gene for which the function or association with disease is known. In other words, is it part of a gene family? You might ask where that gene is actually expressed, in what tissues. Is it expressed in the brain? Is it expressed in the liver? Is it expressed in a tissue or organ that's relevant to my disease? And what happens if you knock this gene out in animal models? Does it change the phenotype? And so there might be studies out in animals that demonstrate a link between this gene and a particular disease or trait. There are databases such as the NCBI Entrez Gene Server, which you can search, for example, for a gene GRIN3A. I just plug that gene in, and it'll pull up all kinds of information, including, for example, that this gene is part of a superfamily. The suspected function it's involved in the central nervous system. There's a knockout mouse that suggests it's involved in development of synaptic elements, and so on. So there's a rich trove of information that you can access in these databases as well. But really, the key, especially when you have a research candidate, and so many of you know, is to find other patients who have this disease and hopefully a mutation in the same gene as yours. And this is really what's going to provide the definitive evidence that this is the gene underlying the disease in your patient. And for that, we fortunately have resources that have come online in the last couple of years, such as MyGene, I guess it's MyGene Squared, which are free searchable databases where patients can go and share their information in the hopes of finding other patients like them. So that's a really great resource. And then, of course, you want to connect with other researchers and clinicians as well. And for that, there are a host of resources such as Genome Connect, Gene Matcher, Peer. You may have heard Ryan Taft and others talk about Matchmaker Exchange in one of the plenary talks. This is a great resource that's come online relatively recently, which is trying to sort of consolidate all of these sources through one search engine. So a lot of different resources out there for patients to share their data, locate clinicians, locate researchers, locate other patients. So I just want, I have 2 more slides. This one, I just want to talk for 1 minute about the success rates that we've been having for diagnostic sequencing. So success rates have been on the order of 20 to 50%, and the rate varies according to whether you use whole genome versus whole exome sequencing, whether you've had any prior genetic testing done to rule out some other genes, and what the disease is. I just want to highlight one study by the Forge group. This is a Canadian group that did a large study in 362 families trying to find the genetic basis. And they asked a question about, well, all of these patients, we've seen these patients for years, they've been in the system, we've had genetic testing done. Why is it that we were able to find genes using whole genome or exome sequencing versus standard targeted gene sequencing? And the top 2 reasons had to do with some of the features of Mendelian diseases that were probably not as well appreciated. The first one being genetic heterogeneity, the idea that for any one genetic disease, there may be many different genes, mutations in many different genes that individually can lead to that same disease. And the other top feature here is atypical presentation. This is the idea that these diseases, the descriptions that you have in databases like OMIM may be based on a handful of patients, and we describe what their clinical symptoms are, and yet we don't appreciate that mutations in this gene can lead to a spectrum of phenotypes or traits, and that you may simply have an atypical presentation and not appreciate that it's part of a certain disease when you're diagnosing your patient at first. So finally, I just want to say that we are still, even though diagnostic sequencing took off in about 2010, we're still in the very early stages of this. A lot of research is going on. We know that there are about 3,000 genes in the OMIM database associated with Mendelian diseases. There are 22,000 genes in the human genome, so there's still a lot of discovery to go that needs to happen to understand what those other genes are doing and if they're related to disease. We've had some really great examples of successes using patient matching. If you've heard about the ngli1.org or adcy.org, these are great examples of patients coming together, finding other patients around the world. And then just for those of you who've had exome sequencing or genome sequencing done, and you've had indeterminate results, couldn't find the gene, check back, because every day we're discovering new genes associated with diseases. So check back, reanalyze your data. So I'm going to turn it over to Caroline now. And I think I'll take questions at the end.
Hi, I'm Caroline Loewy, and coming from a different perspective than Jeanette, she gave an excellent technical background on how to use sequencing and what it all means. And I'd like to give a little perspective from a parent, patient advocate perspective. I'm the mother of an 11-year-old son, James, who after about 9 years of testing and search for a diagnosis was finally diagnosed with a very rare KCNQ2 mutation using whole exome sequencing back in 2013. So after going through years and years of testing, we're finally able to understand the cause of the seizures he'd had since his first day of life and his severe global developmental delays using that sequencing. So in the search for a diagnosis, obviously sequencing is just one of many potential approaches. Historically, clinical evaluation was key, and certainly in our path, that was one of the main approaches. Even in spending a week at the NIH Undiagnosed Diseases program, that full week was largely focused on clinical evaluation. And I think that unfortunately is still what a lot of time on that diagnostic journey is spent in that approach. I think both because of concerns about cost from insurers of moving on to sequencing and potentially what the utility might be of having a sequence, particularly in cases where there may not be a lot of patients and where there may not be treatments for whatever disorder they have. Single gene tests, which Jeanette mentioned, we went through probably 100 of those, and obviously sequencing not available some years ago, but I do think that those end up ultimately kind of overestimating the ability of experts to pinpoint which specific disorder to test for, and ultimately end up being a lot more costly and time-consuming than going more rapidly to sequencing. But obviously the information that you generate through the diagnostic process really impacts what kind of information you might get going forward. So Jeanette touched on this, but just to this has really been an important point to me as I've dealt with many undiagnosed patients and many patients who presumably have a diagnosis, although don't have a real cause. The key difference between genotypic and phenotypic diagnosis and sequencing really is the key differentiator there. The genotype, as I think Jeanette referred to, is the set of genes in our DNA that are responsible for a certain trait, or in these cases, a certain disease. The phenotype is that physical expression, really that collection of symptoms. And that's really an important difference when you think about what can you do with the information that you get from that diagnosis, of which one of those is it. So, what's in that name? Historically, the diseases were labeled based on usually the doctor that found a number of patients, described the characteristics of those patients, and then put his name on the disease. In the case of my son James's disorder, there's many of the patients that were diagnosed initially with Odahara syndrome, which was this collection of symptoms, seizures the first day of life, and severe global delays. But in sequencing, we found that as many as 20% of those patients actually have this KCNQ2 mutation that's causing their disease. The others probably have some other yet to be discovered genetic mutation, and that's true across the board of all sorts of different disorders. Many of the patients that now have come to be diagnosed with KCNQ2 were diagnosed with cerebral palsy. There's all sorts of these phenotypic diagnoses that are useful in describing the disease, but not so useful if you think about moving forward, looking for treatments, and what you can do in understanding the disease better. That's possible with sequencing. The one kind of result of having these better diagnostics available with sequencing is you have newer, rarer diseases, fewer patients in each category, and new diseases that people are not familiar with. When James was diagnosed with KCNQ2 in 2013, there were only a handful of patients that had that diagnosis. The gene was really only characterized in 2012, so 1 year earlier. And some of the cases, the NGLY1 story that Jeanette referred to, initially a single patient that was able to reach out and start gathering patients. And so, what do you do in that situation? And I think that building a community and accessing the community, the sequencing information, working through some of those matching programs and databases, but even the low tech as a parent and patient advocate, working through social media, outreaching, and building a community around that we now, for KCNQ2, have over 200 families in a support group on Facebook, have formed a foundation and have been able to increase the awareness around this disorder. In forming the foundation, we now have an annual awareness week, reaching out and educating experts, and an annual medical and patient summit that this year has about 160 attendees. So in only a couple of years have made huge strides in building that awareness, and really as a result of identifying this gene and the ability to connect the thought leaders. So there's a number of experts. In this case, it's a potassium channel, It's involved with epilepsy and neurology, and finding the experts that have interest in that and having the gene and a target that they can work on has been really critical in getting the involvement of the experts around that. As I mentioned, having the symposium where people meet face-to-face, see the patients in person, and that's been one of the key issues with rare diseases is most experts see maybe one patient. If you're in research, you've probably seen no patients. And the ability to gather up that phenotypic data, be able to publish journal articles that then make their way into these databases so you have better diagnosis through subsequent sequencing of additional patients that they're aware that this KCNQ2 mutation exists. And that's what's really driven a building of the community and has made the diagnosis more useful so that now we can actually do things with it. So in terms of driving research, now that we have critical mass of patients and that we have this genotypic diagnosis, it's really been a foundation for advancing research. We've been able to get grants for establishing mouse models, so we have animal models of our disease on which potentially targeted therapies can be developed. We've partnered with a biopharmaceutical company that's developing cell lines and screening existing drugs against cell functional models of our cell line for a KCNQ2 mutation. And it's the opportunity, hopefully, to get really targeted gene therapies at some point, which are only possible when you know what gene is responsible and what might be a curative approach to treating the disease. And really, even without all of those efforts that are going on the more medical, community, scientific side that we're acting, I feel like the sequencing, at least for us, has really enabled us to better understand the patient. Because for a child that can't talk, that can't understand language, there's no real explanation for what's going on inside his head, why he has certain behaviors or does certain things. And no real prognosis. And so that mechanism, even before finding any patients that are older or seeing what that path is, knowing the mechanism, the underlying gene, has made a critical difference in knowing what his life expectation might be, knowing what other family members might be affected, and it's made a huge difference in just the day-to-day of what therapies might be useful based on how his neurons are functioning because of this mutation. So I think for anyone approaching sequencing and understanding what the utility is, the idea that there's not a specific treatment available as a reason for not pursuing that specific genotypic diagnosis through something like sequencing, I think is really limiting and not understanding what the potential is here and what you can do with that information. So lastly, just the final slide before opening it up for questions. I just want to leave with the understanding of this really brings hope, and in line with the Global Genes mission of hope, it's really empowering to know what the target is, have something to work towards. I've been involved with Global Genes in the rare disease community for some time, but I didn't have anything to work on for my son's specific disorder until we got that diagnosis and knew what the exact cause was for his disorder, and having the hope that goes along with being able to work towards something that might be beneficial to him has really been a critical outcome of gaining that information and understanding. So with that, I'll open it for questions, and Jeanette, if you want to come up.
Yes, thank you guys so much. And like Caroline said, I've been with Global Genes for about 5 years, and the sequencing has changed so drastically from year to year, and as you guys probably well know. So yeah, we have about 15 more minutes. So if you guys would like to ask questions, feel free. This is really for you. So.
Sure. Hi, thank you so much for talking and enlightening us all. My son was diagnosed with a SHANK3 mutation 2 years ago via whole exome sequencing, and I'm wondering, is there any reason why I should want to or push for whole genome sequencing as well?
answer, right? So he had a definitive pathogenic mutation in a gene. So is there a reason to look at whole exome or genome? I would suspect not at this point. Was it a gene that was known to cause any disease, or is it okay? So in that case, I don't really see any value in undergoing whole exome or genome at that point.
Well, it was discovered via whole exome. So I'm wondering.
Oh, okay.
It was discovered via whole exome.
And you want to know if you want to do whole genome now?
Yes.
Yeah, I would suspect not. And for some of the reasons I mentioned earlier, for Mendelian diseases, most of the known variants are actually going to be in the exome and not in the genome, in the rest of the genome, non-coding. Mhm.
Thank you. Thanks very much for the talks. I'm Gary Davis. I'm the manager of Cell Design Studios at Millipore Sigma. We make genetic models of cell line models of genetic diseases. A question for Jeanette, I guess, and that is, can you speak to something that's starting to concern me, is that mosaicism and chimericism in the population, and how that might confound any kind of genetic analysis at the genomic level. And it sort of begs the question, should people be testing and getting multiple tissues sequenced from an individual to be sure that they're getting the correct genetic diagnosis?
So you bring up a good question about mosaicism and chimerism. So basically, just to put it in perspective, when you have what we call a germline mutation, in other words, a mutation in every single cell in your body, it typically arose in the sperm or egg. When you have, after embryo is formed and it's in the early stages of cell division, if the mutation happens to occur then, you can have certain tissues or cells in your body with the mutation and others without, and that results in sort of a mosaic. And so maybe there are some complete organs that have the mutation and others that lose it. And you wouldn't detect that necessarily when you do whole genome or exome sequencing. And I think it's being increasingly appreciated that these are contributing, this mechanism is contributing to diseases. And as far as going back then and testing, I'm not sure how common it is now. If we had any genetic counselors in the audience who might be able to answer that question. Do you have experience, Erica, with a lot of mosaicism or chimerism in your practice?
There are definitely some cases of certain types of disorders where it's fairly common, and a lot of times one of the things that we look at is the family history. So sometimes you'll see things like parents having mild symptoms. Oh, thanks. Yeah. I was just looking for it, but Ryan Taft, who just spoke, obviously, actually published a paper on this a couple of years ago on neurologic disease. And so there definitely are some cases, and it really is important for the lab that's doing the testing to be aware that maybe that's a possibility, because our pipelines are very different when we look at mosaic cases. So it's good to have your genetic counselor or physician, let your lab know, or ask the lab how well they pick up mosaicism.
Right, so that was going to be my question, being from Illumina and having all the experience that you have, how well whole exome or genome picks that up, or do you have to specifically know to go look for it versus doing something else above and beyond whole genome or exome?
It varies by lab. Some labs have very specific pipelines when they do and analyze the data that will detect mosaicism, but you can't pick up everything in one pipeline usually. So some labs will have it, some labs won't. But oftentimes you can actually ask your lab to go back to the raw data and look at the data to see if there's any suggestion of mosaicism. So what a lab's able to report out on the clinical report and what the lab may be able to give you hints to separate from that are actually very different.
Thank you. We still have about 10 more minutes, so I will try to get the microphone to you guys right away so that the people being live streamed can hear. I'll come right back to you one.
Hi, I have a son that's undiagnosed. He had exome sequencing in 2013, and we just recently went to the NIH Undiagnosed Disease Program, and they're initiating whole genome. My question, with the exponential growth we know and discovering disease more and more every day, we're looking at privately having my insurance rerun the exome sequencing data through the computers again. What advantage, because I know exome is only 2% of the genome system, since we're looking at the entire genome, how often do we want to keep looking at the exome or just kind of leave it all to the genome? Does that question make sense?
Yeah, that's a question that everybody has. If I'm doing sequencing for this diagnostic dilemma, do I do whole exome or whole genome? And there are drawbacks to both technology. I think I mentioned that most Mendelian disease variants are located in the exome, so you would think, well, maybe there's no reason to look at the whole genome, and yet we know, comparing whole exome to whole genome, you do get a slight increase in diagnostic yield if you go to the whole genome. It's not because those variants are located in the non-exome region, rather it's a question of quality, because when you're doing whole genome sequencing, the quality of the sequence data tends to be much better, because when you're doing whole exome, you're trying to extract out or capture a portion of the genome that's the exome, and those capture methods are not always perfect, they're not always great. So if I were doing it, boy, I would probably start with, if I had a number of candidate genes. If it's a known disease and there are candidate genes out there, I would probably go and test those first, because when you're doing single gene or even gene panels, those are usually enriched in a way that you're getting great quality. So if there's something there, you'll find it. If you don't, then I would probably reflex that out to I would probably do whole genome if it weren't a cost issue, just because of the quality.
Let me just add one other point to that is what I've found also, and from companies that we've worked with, is that who and academic centers is who's doing the interpretation also makes a difference, not just the quality of the actual sequencing. It's still at a point where it's maybe a bit more of an art than a science, and many of the databases, even just the way they're annotated as far as a symptom, if you're doing a search, if you put a word in a different format than it might be in the journal article and things like that, it comes up differently, and they might see something as, of a variant of unknown clinical significance, which someone else who's familiar, more familiar with that specific area, like someone who specializes in rare neurological disorders or rare metabolic disorders, might have a higher probability of identifying something as being a significant variant than someone else.
Hi, so my name is Emil and Santiago Volker, and I just want to give a little bit different perspective from to families who are still searching not only for a diagnosis but also for a cure. So my son was actually, his name is Nick, and he was dying. I'm sorry?
Patient number one, solved with whole genome
He is
sequencing.
patient number one. And so with exome sequencing, they found a 1 in a billion mutation. And then also another rare disease. So he actually had 2 rare diseases. But Nick was really lucky because there was actually a treatment for him. So he's actually cured right now of 2 rare diseases. But he does suffer from complications from everything that he went to prior to getting that sequencing done. And so I think there was a family that was asking originally about whether they should go back. And from my perspective, I've actually gone back and had whole genome sequencing done, just to look for other markers, mainly for epilepsy. And there were a couple of different markers that were found. And then after having it reanalyzed, because technology and data keeps changing so quickly, other things have been found as the years have gone by. So I just wanted to, hopefully share that information to give hope for people who don't have a diagnosis for their child or their loved one right now. And then secondly, maybe to keep going with reanalyzing if there's any chance.
You bring up a really great point, and I think you're right on in terms of if you have a case where not all of the symptoms that you're seeing, not all of the traits that you're seeing are accounted for by the gene that was, putatively underlying your disease, that it might make sense to go back if you want to uncover possibly a second genetic variant, which doesn't happen very often, but in your case, right, you're one of the outliers there. It can happen where you have actually the misfortune of having two genetic diseases at once, causing this constellation of symptoms that isn't one disease, it's a couple of different diseases. Who's got the mic?
Hi, my name is Aaron Harding with the SYNGAP Foundation. So a question is, Nextgen sequencing is sort of the standard, diagnostic tool, but that we still have microarray. Can you comment on the differences, again, historically using microarray, now we're converting to Nextgen sequencing. Those who have been diagnosed with microarray, what's the comparative, your experience with maybe the comparison between older diagnostic tools, microarray, to Nextgen sequencing, and somebody who was diagnosed with microarray, should they be reanalyzed with Nextgen sequencing? And then also another comment, as I work in the laboratory field doing requirements, working with the providers on what genetic requirements they're going after. And the problem within the genetics is standardization. You can go to several different laboratories and they're not standardized. And I think being a laboratorian, we have to work within the community of standardizing tests across laboratories based on literature, evidence-based, to hopefully get to a point where we're speaking off the same sheet of music.
So to address your first point about microarrays, remember the slide I showed you with the different variants in the human genome, and the most common are these small genetic changes, but you can sometimes have these large genetic changes, like big chunks of DNA that are duplicated or deleted. Those are called structural variants. And when you do a microarray, it's a powerful approach for looking for structural variants, which the large ones are very deleterious. We also all have a bunch of relatively small ones, but those are not picked up very well. The large ones are not picked up with whole genome sequencing. So even though we say whole genome sequencing, it's really specialized at finding these small genetic variants. That's in part due to the technology right now, which is really relying on small stretches, like 50 to 100 nucleotides of DNA that we're sequencing and kind of piling on top of each other. As the newer generations of sequencing technologies come out, we'll be much more capable of detecting structural variants with whole genome sequencing. But for now, microarrays are the standard, gold standard for that. And the second point that you made about standardization, I think there's a real effort now in the community for people to standardize their vocabulary using, there's this human phenotype ontology. It's basically a standard language that people are being encouraged to use in the medical field, where you start with something like, seizures as a broad term, and then it has offshoots of that are very, very standard terminology, where you can actually drill down and get into very specific traits with a standard vocabulary and roll them up into bigger categories. But again, if you have the standard vocabulary, then you can use a database like OMIM and plug in a bunch of terms that everybody knows this is what they are. It's not diseases of the eye, it's eye diseases type of thing. That's where we're going right now, and that is important, definitely.
Thank you. I wonder if you could say something about what's being done to understand the Intron area, if that's a simple question. If there's a big push like there was with the exon area.
So the Intron area, so that's within the gene regions that are actually spliced out and not part of the coding region. They don't end up in the protein. I would group that more in with the whole 98+% of the genome that is non-coding, which is a big mystery now what it's actually doing. There are efforts ongoing to try and figure out what all of that DNA is up to. There's the ENCODE project that the NIH funds, and that's definitely where everyone's focusing their attention, trying to figure out what that region of the genome is doing. We're making some progress. It may be involved in regulating which genes are turned on and off, for example. But yeah, that's definitely a need, right? You got your whole genome, but we don't know what it does.
Actually, I'd like to dovetail right into that. I have a daughter who, after many years, was diagnosed with a very rare disease. We found 3 people in the database who have the same gene defect. And when I look at her report, it says there's a missense. They know which gene it is, but then everything is uncertain and uncertain and uncertain. Where does this lead us? What is research doing? How can you find out what does the protein do? Does it turn on or off, or more or less?
Right, so just because you have a gene that is you have a mutation in a gene that's known to cause disease doesn't mean that that mutation causes disease as well, because we all have lots of mutations in all of our genes that are doing absolutely, nothing to the protein. So figuring out the pathogenicity of variants is something that everyone's really focused on now, and we use a lot of different pieces of information. Some of that information's not available, though. For example, animal models. I showed you the picture of a mouse. You knock the gene out, maybe you knock that specific variant out and see how it affects a trait in an animal. Those types of experiments aren't done on a large scale unless you happen to find someone researching that gene. Otherwise, the best thing that can happen is for people to share their data, and you find other patients who have that same variant and with that same disease, and everyone knows about it, and then we can make a more definitive diagnostic.
Hi, my son has a clinical diagnosis of FG syndrome. It's a developmental disability, and the clinical diagnosis is of a certain type where they don't know where it's located exactly. So we have that, but then we're also seeing a metabolic geneticist, because we know he has primary lactic acidemia, and we're trying to see if he has something metabolic or mitochondrial in addition. So we're undergoing whole genome sequencing. Someone else is doing whole exome, but that's for the research side of things. So there's so much going on. So anyways, when this whole genome sequencing comes back in about a month and a half, I think they're kind of hoping to prove the FG syndrome and then see if anything else is going on. But I'm kind of afraid that we lose. They'll say, well, we found no, proof of the FG syndrome, and then I don't know what they'll find metabolic-wise. So, having an answer, that one part means a whole lot to us as far as that clinical diagnosis. So I don't know what I'm looking for here, but do you have, I guess we've kind of touched on it, but any advice for someone in the situation like we are, or who's looking at the possibility of 2 rare disorders and maybe of a different type too?
So you're worried about getting the results back and not finding anything, or?
Perhaps, and then maybe losing the clinical diagnosis, the one really good one we do have, which I feel is thank you. Losing the clinical diagnosis, which is a great fit, frankly. I want an answer, and I want the right answer, but I don't want to go from having something to all of a sudden being back in no man's land again.
How solid was the first diagnosis?
It's a clinical diagnosis. So it's very, very convincing.
But you had the gene. Did they find the genetic variant already?
There's 7 types, and they think he has the one type where they don't know the gene, so they initially said don't even test for that, but now they're they're kind of hoping they find it there anyway. It's very convoluted, but we've got a couple people working on this from different ends, but right. I don't know what I'm looking for. I just didn't know if you had any advice.
You bring up an important point that my medical geneticist colleagues always remind me of. The most important part of communicating with patients about whole genome and exome sequencing is upfront, before you get it done, to let them know that uncertainty is the norm. Uncertainty is part of the game. And again, if you have a variant, even in a diseased gene, it still could be called a variant of uncertain significance. So just to brace yourself for that, but hopefully more information will come along in the years to help clarify that.
Thank you.
Okay, I think we have time for one more question.
You've talked about the N of 1, and sometimes I'd like for you to discuss how my daughter has a disease that is not an N of 1. There's probably about 15,000 pediatrics and 50 to 100,000 adults. But the N of 1 comes into play because she is on an experimental treatment using an orphan drug on an off-label use, and it's reversed all indications of the disease. But physicians around the country and around the world refuse to use the treatment, even though it's worked for her, and there's probably 13 patients now at Stanford and maybe 30 patients at Mayo Clinic. But the N of 1 is, well, there's been no clinical study, and there's no clinical study because there's no money to study generic drugs, and there's no data, so there's no money, and there's no money, so there's no data. And so, you know, I mean, there's probably there's probably 31 loci and 34 independent associations between, you know, ulcerative colitis, this disease, primary sclerosing cholangitis, and then a number of other autoimmune diseases. And so at some point you have to go, maybe you go forward with patients who, you know, you take all these N's of 1, put them together, and then have to do reverse engineering to see why is it that certain patients respond to this medication, why is it that others don't, and how do these genetic associations, you know, how does genome sequencing work for these patients? You know, I mean, it's so complicated because it's not just one gene and it's not 2 genes.
Right. Did you want to address that at all, your experience?
Sure. I mean, and I actually, as a sideline to being a parent of a child with a rare disorder, also work in the finance area in the biopharmaceutical area. So I've, you know, done a bunch of work professionally in that area in small companies, but you know, the approaches, I mean, to drive off-label use typically is publication of the data, is what in a peer-reviewed journal. So if there are these, not just N of 1, but 10 patients here, 10 patients there, having, you know, pushing and supporting any efforts to get that published in a relevant peer-reviewed scientific journal will allow more use off-label, particularly if it's a relatively inexpensive generic drug that gives other physicians in other places more comfort with prescribing it off-label, what the dosing regimen is, what side effects might have been seen, you know, any kind of data on what the mechanism might be. So, I mean, that's certainly useful in that regard to try to get off-label use. And there's also opportunities working through the NIH, especially, you know, the fact it's a generic drug is fabulous in terms of anyone can acquire the drug. The NIH, you know, pushing for the NIH to sponsor a trial and activities like that are certainly
under the
avenues.
Orphan Drug Act, but we have filed a PCORI grant, so we're hoping to hear from that. And we're going to foreign investing, so it's.
You also might want to talk with Gay Grossman, who is here. She's working with Nick Shork in San Diego on N of 1 trials, right, Gay? So maybe catch her at the break and she can fill you in on what they're doing.
Okay, thank you both so much. Thank you guys for your incredible questions. We have about 10 minutes until the next session. And if you're staying in here, great. But thank you so much. Thank you. Thank you.